Now let us discuss the next step in our DNA technology, that is ligation. So we have discussed isolation of gene of interest, then the fragmentation, separation using gel electrophoresis, amplification using PCR technique. And now the next step is ligation. What's the meaning of ligation? Now we have enough number of desired gene. Now we are going to ligate it or join it to the vector, or sometimes directly to the host DNA. So that is the next step. So for that, what we should do? So desired gene is there, the vector or the host gene. Sometimes we give it to vector, otherwise we are directly giving it to the host cell. So based on that, vector DNA or the host DNA is taken. Then DNA ligase enzyme is added to it and it is incubated. What's the meaning of incubated? Enzymes require optimal temperature and all other conditions for their action. So high temperature and low temperature can make it denatured because they are all proteins. So maintaining all the optimum temperature and all is called as incubation. So when it is incubated, what will happen? This will join and form the recombinant DNA. Now the next step is transfer of rDNA. Now here we have made, so ligation is a step of actual making of rDNA, and now we are going to transfer the rDNA. There are two methods of transfer. One is direct method or direct transfer. The other one is indirect method or the indirect transfer. Indirect transfer, there are different methods based on the host organism. If E. coli is the host organism, how can we transfer the rDNA by direct method? Two methods are there. One method so the first method or the method which we need to study is chemical method or chemical treatment and heat shock. This DNA is a large molecule and the membrane is lipophilic in nature. You have learned it last year. It's made up of lipids. So first for lipids cell membrane undakirikkunnu. So water loving substances cannot easily pass through the cell membrane, and the cell membrane pore size is also very small. So DNA is a large molecule. It cannot enter through or it cannot pass through the membrane pores of the cell membrane. So we have to make it competent, or we have to make it in a state which is ready to accept or to get that rDNA. So for that we do chemical treatment with divalent cations like calcium and all, Ca2+. So it is treated with the cell, that E. coli cells are treated with calcium chloride, and then it is incubated along with the rDNA and heat shock. First it is placed in ice, that is the E. coli bacteria which is already treated with calcium chloride to increase the pore size is now kept in ice along with the rDNA, and then it is transferred to 42 degrees Celsius, that is a higher temperature, for a few minutes. Again they are transferred to ice. Like that we give heat shock treatment. So by this what happens is that the rDNA will enter the cell because the pore size will increase by this method. Another method is, we need not study, but it's better to know this, electroporation. Either E. coli cells electric field the size of the cell membrane, the pore size of the cell membrane will increase, and this will also help to take in the DNA fragments. So this is the direct method in E. coli. Now if animal cell is the host organism and if we are going to give it by direct method, we use a method called as micro injection, where glass made micro needles are used or micro pipettes are used, very tiny small things, and by which we are directly injecting the rDNA into the nucleus of the cell. And this can be done, micro injection is possible only in animal cell. If plant cell is the host organism, we use a method called as gene gun method or biolistics, where there is a machine which can shoot the rDNA inside the nucleus. For that, this rDNA is coated on tiny microparticles of gold and tungsten, and it is hit into or bombarded into the nucleus of the plant cell with high velocity. So the gold particles, microparticles of gold and tungsten is coated with the rDNA and it is bombarded into the or shot with high velocity into the nucleus of the plant cell. So if plant cell is the host cell, the direct method is gene gun method or biologistics. Now indirect method. What are the various indirect methods? Indirect methods plants and animals indirect method is by using disarmed pathogens. We have already learned about the natural genetic engineers, that is agrobacterium tumefaciens in dicoplants and retroviruses in animal cells. So they are disarmed, that is pathogenic genes are removed, they are made non-pathogenic and the gene of interest is added to them and they are made to infect the animal or the plant cell. Thereby, it's an indirect method of transferring the gene to plant or animal viruses. By indirect method in bacteria, bacteriophages can be used. Okay, so that is the indirect method. So these are the ways by which the rDNA is transferred to the vector or directly to the host organs.